Hello everyone. Welcome back. This is lecture 11 now. The last couple of weeks we've been talking about how to construct a within host vital dynamics model. We're gonna review some of the steps that were discussed last class, specifically in terms of how to include immune modeling. What we will be doing today is that we will take the model and we will, because we constructed the model with the explicit intention of having it fit the specific phases of the vital load that were collected by Amber Smith. And now in this class, we will compare the model that we constructed with our initial parameter estimates to the data, and then we will try to fit the model to the data to get a better estimate of the parameters. And then that's gonna be the first half of the class. Then we'll take a break, grab coffee, we'll come back, and then we will spend some time trying to replicate two different papers on within host vital modeling. One has to do with hepatitis B vital dynamics, and the other one is one that models mutation of HIV, and we will talk a little bit about those models. Okay, so as I mentioned, those are the topics that we will be covering today. Fitting the within host model to the experimental data and replicating two papers. So last time we spent the last 2 or 3 weeks constructing what it's called the standard kinetic vital model. The model usually has cells of 3 or 4 different types, depending on what the model is what information the model is interested in tracking. It has cells on an uninfected state, cells in a early infected or eclipse phase state where they are infected but they are yet not producing virus. Cells in a productive state where they are infected and produce virus. And then these late infected cells die because of viral production. And then this virus that is produced by the infected cells contributes to further infection of uninfected cells. And that virus can also decay. The way to think about it is that the virus is released to the extracellular environment, and then the virus either propagates through the tissue and infects other cells, or it can also be degraded by for multiple reasons, including some aspects of the immune response. So the rates included in this, the rates that describe this vital kinetic model are the infection rate that is determined by this parameter beta that we've seen multiple times. Is the infectivity of the virus. The eclipse phase rate coefficient that determines how long the cell spends in this eclipse phase, and we saw that can vary for different viruses and even for the same viruses for different viral strains. But typically in influenza is about 6 hours. SARS-CoV-2 can be up to 12 hours. And then this infected cell clearance, it might represent multiple different mechanisms. In particular, it can sometimes represent cell death due to viral production, cells sensing viral production and then inducing apoptosis and dying. But it can also represent the effect of the innate immune system, the response to the response of the immune system, including some of these infected cells. And we talked last week about some of the mechanisms that the innate immune system can use to clear some of these infected cells originally. And then this model usually assumes that infected cells produce virus at a constant rate, that it's called P here. This might be an appropriate assumption under certain circumstances. We worked more complicated models. Some of the models we were looking at over the weekend have usually can integrate two different scales. Let's say as a modeling of at this level with a model that represents reproduction of virus inside the cell, and then make that production a function of other variables. But in this model, it is assumed that it's constant. Okay. So we did talk a little bit. So as I mentioned, the clearance in this model is usually represents the activity, the innate immune response. And that allows us to represent the vital load curve up to day 8. But what is observed in this vital load curve is that after day 8, when the adaptive immune system kicks in, there is a rapid clearance of the virus. So to be able to reproduce that rapid clearance, we need to include the adaptive immune response in our model. And in the actual biological systems, the usual way that the adaptive immune system is stimulated, it's either by secretion of certain inflammatory molecules or signals, or by the transport of certain messenger cells from the infection site to certain lymph node organs, where they actually recruit and induce proliferation and differentiation of the cells associated with the adaptive immune response. So having that general idea of the transport between infection site and lymph node organ, and what happens at the lymph node organ in terms of specific cell differentiation, and then the transport back from the lymph node to the infection site, allows us to come up with a model. Having that in mind, we can come up with a model that includes the different steps in this stimulation process. And again, the idea is that once we include those different steps, we should be able to see or reproduce a rapid clearance of the vital load when the adaptive immune system kicks in. So the first thing we need to include in our model that we're missing is this cytokine signal, or let's just call it in general a pro-inflammatory signal that needs to be transported from the infection site to the lymph node organ. And as James mentioned last time, we used a unfortunate word name. We chose an unfortunate name for the signal because we just restricted it to the cytokine signal, which is it assumes that it's an actual molecule that is being secreted by the by the infected cells. But in reality, the stimulation, as I mentioned, occurs in two different ways, both by the secretion of these molecules, but also by some cells that, some specific cell type that connects the innate and the adaptive immune response, transporting information from the infection site to the lymph node. But in general, we'll start this model by adding another level of complexity in which our infected cells secrete this signal that we're calling cytokine. Again, what we do is that we have our cells secrete the cytokine at a constant rate, and then this cytokine, same as the virus, can degrade in the extracellular environment because of instability of the molecules. And in the case of cells, some of the cells that actually transport this information can get infected themselves and they can die. So we can think of that as a signal decaying after being released by the infected cells. So we included the cytokine production and decay in our original model with a constant rate of production by both early and late infected cells, and then a decay rate. And then what we see is that our cytokine curve mimics the vital load curve with two specific differences. There is a small difference here at the beginning, and then a difference here on the amounts. Can someone think where these differences are coming from? Like, any ideas? So it's clear what I'm asking. I'm asking, once I include this, right, both my early infected and late infected cells produce this cytokine, and that cytokine decays, and that curve mimics closely the vital load curve. So at least we know that here the difference has to do with the fact that as soon as my cells get infected, they start releasing the cytokine signal, right? Because in the virus, they have to spend some time in that eclipse phase where they're not releasing virus, and then they start releasing virus later on, right? But for the cytokine, they start releasing as soon as they get infected, and that is the delay that we're seeing here between the cytokine and the virus. And then the difference here has to do with multiple things, right? It has to do with the production rate, but the production rate is quite similar between them. Has to do with the decay rate, and we see that the virus decays much faster than the cytokine in our model. So you would expect to have a bit less, everything else being equal. And ultimately, the fact that you have more cells secreting cytokine that you have secreting virus, because both cells in the early and late infected secrete cytokine, as opposed to just cells in the late infected phase that just secrete virus. Okay, is that clear?
Yes.
Okay, so, and then I talked a little bit about this already. These are the type of cells that transport the information from the infection site to the lymph node organs. It's called sentinel cells, specifically dendritic cells and macrophages. They basically they phagocytose parts of the virus from the extracellular environment, or they collect information from cells that are infected, and by recognizing certain cell surface receptors that indicates that the cell is infected, and they transport this information to the lymph node, basically collecting the ID of the virus, and then going to the lymph node and reporting, okay, this is the virus that it's present in tissue. So I need effector cells. I need cells of the adaptive immune system that can actually target this virus, basically profiling the virus. So this is important because in our model, in the first model we constructed it thinking just in terms of the cytokine signal being transported to the lymph node. But in reality, it's a combination of, yes, pro-inflammatory signaling molecules, but in terms of the specificity for differentiation, that is stimulated by the information collected by these sentinel cells. So now that we have the cytokine signal produced in the tissue, we can transport this signal to the lymph node. And this we do by what is called a transport equation, where we basically say, okay, a fraction of my signal leaves the tissue and then appears in the lymph node. And that transport rate is given by the transport parameter, which we're calling here Kc. And then once in the lymph node, that transport parameter, of course, it depends, like how long does it take for the signal to reach the target organ, depends on where the infection is happening, what type of infection is, whether it's transported through the lymphatic system or it's transported through the blood. So there's multiple parameters that control that rate. Here we assume that the APCs travel to the lymph node in about 2 days. It takes 2 days to reach the lymph node. And then once the signal is at the lymph node, it takes 2 days for the cell, for the signal to decay. So we included in our system by having these transitions, right? By the cytokine in the tissue going to the lymph node, that's what we call a CL, and the cytokine in the lymph node eventually decay. And then when we add that, we saw that now we have a little bit of a different relation between the. So I'm sorry. So we have the cytokine signal, right, activated as it was in the previous part, but we also now have a cytokine in the lymph node that that mimics, but it's not exactly the same as the one in the tissue. And again, the difference here, what is a good guess of what is determining this difference? So this is telling me how much of the cytokine I have in the lymph node. And this is telling me how much of the cytokine I have in the tissue. And I said there is a parameter, the transport parameter, that determines how long does it take for my cytokine to go from the tissue to the lymph node. And that is the one that's going to control the distance between these two, right? Because if the time it takes is big, then we should see a bigger delay between the signal in the tissue and a signal in the cytokine, in the lymph node, right? Does that make sense? Okay. So once the signal reaches the lymph node, then it induces the differentiation and proliferation of certain specific cells of the innate immune system, of the adaptive immune system, I'm sorry. And what is special about this type of cells is that they are really good at identifying which are the cells that are infected and inducing cell death in those infected cells. So, and that is why it's called specific, right? Because it's able to recognize and distinguish the cells that are infected by the virus because the information was passed on by the sentinel cells in the lymph node. So once the signal arrives to the lymph node, then it can induce the proliferation of this specific cytotoxic cells. Cytotoxic means that they kill other cells. And we spent a little bit of time talking about the meaning of this equation. I'm going to spend just a couple of minutes explaining what it means. So there's two things that need to happen in the lymph node. The cells, these effector cells need to proliferate. But for them to proliferate, there has to be some of them, right? If you have none, you cannot proliferate from there, right? So there has to be an activation where you produce some of this or activate some of these effector cells, and then once you activate them, then they can start proliferating. So, and these two terms here are accounting for those two different things. The first term is the activation of the effector cells, and that activation, you guys see, is only dependent on the amount of cytokine that you have in the lymph node. So once the cytokine arrives in the lymph node, or the signal arrives in the lymph node, then it activates some of these effector cells. And then the other term is a bit more complicated, but basically, you see that it's dependent on the number of effector cells. So that implies that the rate of increase of the effector cells is dependent on the number of effector cells that I have, which means that they're replicating. But they're replicating only in the presence of cytokine. So if you don't have any cytokine, there is no proliferation. So notice that cytokine is needed for both activation of the effector cells, but also for proliferation. And the proliferation rate is determined by this parameter, R. And then James spent a little bit of time explaining the importance of these type of equations, what we call the saturating functions. And the idea of this function is that there is a limit of how much effector cells you can produce, and that limit is reached when the effector cells are high enough. So, and what high means is determined by this parameter. So we said that it's 1,000 cells. After you have 1,000 cells in the effector cells in the lymph node, then their proliferation rate starts decreasing. Because in that case, this number is high, so this terms becomes 0. Okay, it's a bit sophisticated, but it's a sophisticated way of modeling the fact that you cannot have infinite number of effector cells. So you need to have some limit on their growth, and this is the way that we model it. It's not the only one though. There's other ways we can model that limitation of the growth, and there is specifically one of the papers that we're going to be replicating today. There is a logistic growth function that is also intended to include that limitation in how much the cells can replicate. So this is important because it's going to come out later. So now we have another component of model. We have these what we're calling our T cells, and we see that our T cells start replicating after the cytokine signal comes in. If the cytokine signal is 0, they do not replicate. So it's dependent on the cytokine signal reaching the lymph node. Then we need to put this back into the tissue, because that's what these effector cells do. Once they proliferate and differentiate in the tissue, then in the lymph node they are transported back. James, and I'm not sure, is there a preferred method to be transferred through blood or the lymphatic system?
I think both. Transfer through blood is faster.
Right. So the same way we modeled, right, the transport of the cells from the cytokine signal from the tissue to the lymph node, now we can model the transport back of the T cells from the lymph node to the tissue. And now that I look at the parameter values, we basically assume that it was the same. So we assume that the same time that it takes for the signal to reach the lymph node is the same time that takes the cells to travel from the lymph node to the infection site. That might or might not be true. I think it's an assumption that we included in our model. And then we also assume that once these cells reach the tissue, they spend about a day in the tissue before dying. So they have an effective activity, like an effective lifespan of a day once they're in the tissue. There's like complicated reasons because of why that might be the case. Some effector cells can actually get infected and die. But in general, we wanted to model the fact that these cells have a limited time where they were effective at targeting and killing infected cells. So that survival time allows us to represent that limitation in the effectiveness of the cells once they've arrived into the tissue. So now we can compare those two. So now we have the cells in our tissue, we have the vital load, we have the cytokine in the tissue and then transported to the lymph node. Then we have the T cells in the lymph node, and then the T cells in the tissue. And by now we should know a little bit what's determining why there is a delay between the lymph number of T cells in the lymph node and the number of T cells in the tissue. And it's the same reason why there is a delay in here as well, right? Because it takes time for the cells to travel from the lymph node once they start proliferating to the tissue. James, another question, because the ratio here between these two it's just a result of the parameter values that we chose, really. What the ratio is between the tissue and the how many cells you have in the tissue as opposed to the lymph node, that is actually something you can measure experimentally. But we don't have that data, so we were estimating the parameters independently, and this is the ratio that we got from the parameters that we chose. If you had data that you could estimate those parameters independently, then that would allow you to calibrate your model better, because you would have a better sense of, okay, what is the ratio of T cells in the tissue versus the lymph node. And we, in the cancer model that we published recently, although it's special, but we do use some of that data to calibrate like how much cells I have to have in a specific location, and that is good to calibrate the model. So lastly, what we need to add is this T cells being able of doing something once they are in the tissue. The way we included it is that when these effector cells, T cells, come in contact with the infected cells, they induce cell death. And now I'm realizing, yeah, it's kind of interesting, James, because we just assume that effector cells can kill cells just that they're at the productive state. But I'm pretty sure effector cells can also kill cells in the eclipse phase, because cells start signaling that they're infected way before they start actually releasing virus. So that is an interesting addition to the model that you guys can, or we can play with it maybe later on during the class. See how the model behaves when the idea is that instead of effector cells just killing the late infected cells, they could potentially also kill the early infected ones, because these cells, the way they operate is that they basically they scan the tissue, and then by looking at specific molecules that the cells are displaying on their surfaces, and when they detect that they recognize the molecules associated with the virus, then they induce apoptosis or cell death on those cells that they recognize as infected. But cells can displace those molecules in their surfaces even before they are already producing virus and releasing virus to the environment. So that's an addition that could be added to the model. In terms of the mathematical form that we chose, we did spend a little bit of time going over this last time. I don't know, James, do you want to go over it again? No. So, but the idea again is that the reason we included this specific mathematical form, it's because we wanted the rate of killing be limited both when you have small number of infected cells and when you have small number of T cells. If you have small number of infected cells, then it becomes harder for the T cells to find them. And then if you have small number of infected of T cells, they can just kill at a particular rate, because they cannot kill faster than they encounter cells and they induce apoptosis of those cells. So that was the idea of it. And James talked a little bit about how to think about this type of equations in these limiting cases that I'm talking about. But the important is that we can now put the whole model together, and we started with the standard model here, what is called the standard kinetic model. We added the cytokine or the signal, and then we actually developed a multi-compartmental model, because we have now two compartments. We have our tissue compartment and our lymph node compartment. And we decided that we will model a transport of the signal from the tissue compartment to the lymph node compartment. In the lymph node, the signal-induced proliferation of the T cells, and then we have this transport back of the T cells to the tissue compartment, where they induce death of the infected cells. Okay. And we modified the antimony string to include this, which shows the specific values here. To be completely honest, I think those values were mostly estimated by looking at the curve and making sure that they look similar to the experimental curve. So it doesn't necessarily have a lot of biological meaning, in the sense that the values of these parameters were chosen such that the vital load curve that we get from the model looks similar to the vital load curve that we were trying to fit at the end. And the idea is that when we get those parameters right, we can then replicate the 4 or 5 different phases of the vital load curve. We have the eclipse phase, where there is no vital production, the exponential growth phase. Then we have the saturation and lower decay, slow clearance phase that is associated with the innate immune response, and then a rapid clearance phase that is associated with the kick in of the adaptive immune response. And if we go back, so we can start seeing here, let's compare this one, the vital load, with the T cells. So here, this orange curve, is the number of effector T cells in the tissue. You guys can see that by day 4 or 5 is really when you start to see some of those effector T cells in the tissue. And around that time is that the decaying curve here starts deviating from the linear decay that you were seeing to be more, linear in this case is exponentially slower because it's in a log scale, but the slow clearance starts deviating away, and you can start to see a rapid clearance coming on as the cells come in. And then as you reach a maximum number of effector cells, then you reach your highest clearance rate in this rapid clearance phase. So looking at these two, what differences can we can you guys see from just from looking at this to the what is major differences between these two? Let's see.
It might not be a major difference, but it seems like the viral load is put off slightly. Like in the graph on the right, it seems to start right at 0, and the viral load on the left seems to be delayed by I don't know, a few hours.
Right. Yeah, that's interesting. I would say, yeah, but, that has to also do with with the axis that we show, the particular axis that we chose to represent the curves. Because basically what we did is we assumed that for a cliff, because in the actual OD model, depending on your initial conditions, you might have some virus at the beginning, but that virus at the beginning does not count as your real vital load. So we basically set a threshold and say a clip phase means that the virus is below this threshold below this threshold. So basically 10 to the 1 is the one that we set up. And basically, yeah, we are kind of like, but when you look at the 10 to the 1 threshold here and here, it's kind of similar. Not terribly, but yes, we might be actually shifted a little bit in terms of the clip phase. What about the rapid clearance at the end?
It seems like the rapid clearance is maybe a little bit more stretched horizontally.
Right. So in the experimental data, it seems like the clearance kicks in later, but it's much faster. Whereas in our model, it kicks in earlier, but it's kind of like slower, right? And that's why you see that this, what is called this slow clearance phase, lasts longer on the experimental data than in our model. And then the clearance phase happens much faster in the data than it happens in our model.
I have a quick question on that.
Yep.
One thing I noticed is that the graph on the left appears to be more stretched out than the graph on the right. So the graph on the right is a little more compressed. So are we actually seeing a faster decline in the right graph, or is it just based on how everything's been scaled in the image?
Right. Good question. Let's look at this. Around day 6, we have, because the saturation curve, the saturation happens around the same values on the same day.
Right.
So saturation at 10 to the 6 around 1.75 days. It's kind of like the same here. Then we have this linear regime in the decay that in our model lasts until day 6.
Okay.
And in their model, yeah, also day 6 too. But then when you look at where it ends, right? Yeah. We'll compare the data to we'll overlay them. That's why it would be useful. But
okay.
Yeah, it's kind of like hard to. Yeah, I would say that's a good point. It's kind of hard to say by just looking at them like that. But yeah, I would say my first intuition would be there is a slower. It seems that there's a slower clearance here, especially because like by day 8 in the data, there's basically no virus, and then we still have some virus after because we're cutting here.
I guess there's a question. I mean, there are a couple of things. One, there's a problem that the experimental measurements were only done once per day.
Right.
So you can't really see what's happening early on because you only have one data point, which is that value at day 1. So it's the time course, the time measurement for the experiment is too coarse to be able to understand the eclipse phase. For 6 hours, you don't see it in daily measurement. And then on the downslope side, something funny is happening at day 8 because at day 1, 0, 1, 2, 3, 4, 5, 6, 7, the replicas all have fairly similar viral loads. But at day 8, one of the replicas has no virus, and 3 of the replicas still have pretty high levels of virus. And so how do you interpret that? Do you interpret that as a time uncertainty, so that those higher levels really represent measuring at day 7.5 instead of day 8?
Right.
Is the shutdown slower in some animals than other, which is possible? And then at days 9 and 10, the values are essentially 0 again. So the problem is that the temporal resolution is not sufficient to be able to distinguish those things. And because you only have that one point at day 8 when there's uncertainty, you don't know why the different animals had different viral loads on day 8. So it's a little hard to interpret what's happening. If you average those values, you'd get a point that lies right on that black line that she's drawn. But it's not clear that you really should be averaging those values, because if there's a temporal jitter, averaging the values between different time courses may not be meaningful. What you'd like is if somebody measured the viral load every hour, but I think Josh will tell you a little bit about how the viral load has to be measured and why you don't repeat those measurements very much.
Right. Right. But that's a good point. And it's a good point to look at, okay, what is the actual visualization of my data contributing to the effects that I am interpreting from the model and my data. So yeah, that's a good point. And we'll see, because I'm gonna compare them like right away. So yeah, this connects exactly to the conversation we were having. So what is where does this experimental curves come from? There's multiple ways of quantifying vital load curves. But in general, what you want to compare your model to is a vital load curve that specifies the amount of virus as a function of time, right? Usually, these curves are measured using what are called plaque assays, and this is a picture of a plaque assay here. And the idea in this assays is that you have basically a plate with a monolayer of cells, and it's usually confluent, which means that the plate is full with cells. And then what you do is that you have a solution that has the virus in it. We'll talk how that solution is extracted, and then you create certain what is called serial dilutions of that sample, and then you infect multiple of these plates, and then they pour a semi-solid medium on top. The idea being that you don't want the virus to flow freely, because what you want to quantify is spots where the virus was able to replicate. And then when you count those spots, literally count them, then you can count what is called the plaque forming units. So in general, the virus replicates inside the cell lyses, which means basically it explodes and it releases virus, and that virus, because the virus cannot freely move in the environment, it infects the neighboring cells. That's the way the virus propagates through the assay. And then that one also gets infected, lyses, and infects further. And what you end up seeing is those kind of like white spots in your plate that represent areas where the virus was able to replicate. and then you do some calculation, you count those areas, and that is going to give you your how many plaque forming units you had in your sample, and then you can use that to estimate the virus back depending on the dilutions that you made. That part is not that important, but the idea is that you basically take a sample with the virus, put it in this infect some cells, and then count how many plaques you get by infected your plate with that solution. That gives you what is called the PFU, the plaque forming units. But if we look at our data here, the data here is expressing in terms of TCID. The TCID is a different measurement because sometimes the virus doesn't lyse the cells. So if the virus doesn't lyse the cell, you cannot count how many dead cells you have in because cells do not die because of the infection. That happens with certain viruses. Sometimes you don't want to use a solid media, so you let your virus to freely diffuse above your plate, and then what you do instead of counting plaques is you count how many cells got infected in your plate relative to the total number of cells that you had, because you can quantify, and then what you get is the TCID, which is the amount of virus that was in your sample, such that 50% of the cells got infected in the plaque assay, and that is called the TCID 50. But it's basically the same principle, except that you don't necessarily count the plaques. You're counting how many of the cells got infected, and there's multiple ways of detecting that. But yeah, so that's sort of that's how you quantify how much virus you have in your sample. Now, let's talk a little bit quickly about how the sample was collected for this specific data set. Okay, so I go back to the paper and I read the methods, and it's kind of gruesome, but it's kind of cool to read as well. So, right, so the viral infection dose, the TCID 50, was determining using a specific method. That part is not that important. But you can see that they use a specific cell type that they plated, and these are the cells that they infected with the solution, right? So these data points come from mice, and those were intranasally inoculated with an initial known concentration of virus. And then they were weighted at the beginning of the infection, and then they were euthanized when they were moribund. I mean, they were really bad, or they lost 30% of their original starting body weight. And then they have 10 mice per point. So that's all those points that you're seeing here are the 10 mice per data point. So basically they had 120 mice, because it's 10 mice per point per measurement. So what they did then was they euthanized them by asphyxiation with CO2, which is kind of common, better than decapitating them, I think. And then lungs were harvested and then digested with collagenase, and then homogenized by side-inch plunger. I don't even know what that means, but that sounds terrible. Sounds like putting them in a food processor, but I'm not sure. And basically, so basically they're digesting the lungs, and then they centrifuge this mix, and then they get the supernatant, which is the liquid that remains because when you centrifuge the sample, the salt lysates are gonna precipitate, and then you get the virus on the liquid, and then the cell, all the proteins associated with the cells are basically in the bottom. So, and then you take that sample, that liquid, and then that is the liquid that you use to then infect your plaque assays to be able to determine the concentration of virus in the lungs. Again, it's a little bit gruesome, but that's the way it's done. And James was commenting that it's extremely complicated to do as well. Like, it's very time consuming. It's done. And then on top of that, right, so there is a limitation on the number of points that you can get here, because it's determined you need certain number of mice to be able to have statistically meaningful. And then how many times are you killing the mice and then collecting the data? It's kind of, it can get very complicated. So it's, you guys can see how this data collection can be rather complicated. Any questions about this? I actually like talking about all of these experimental. In my other life, I was an experimentalist. Okay, so going back to now the model. So what I did was that although I'm pretty sure we could have contacted Amber to get all the original data, what I did was I digitized this, which gives you the average for each of the samples at each particular time. And then in that link that James shared with you, there is for the file, there is a file called smithdata.txt, and that has the specific time points associated with this curve. Okay, and then there is also a notebook called Vital Kinetic Fit 2. That is the one that we're gonna be using to try to fit our original model to this data. And I want people to load it. I'm gonna create a poll to make sure that everyone, to wait for everyone to be able to launch their Jupyter notebooks and make sure that you load both the data set and the Jupyter notebook. Okay, so just give it 1-2 more minutes to give everyone the chance to upload the Jupyter notebook. Okay, so yep, we can move on. So the first part on this notebook loads all the libraries that we're gonna be using. Some of Tellurium is already familiar with for us. LMFit is the one that we're gonna use to specify parameters and call the optimizers. We'll talk a little bit about that later, but it was also loaded in the previous notebook that I shared with you that I also did some fitting. And then the first part that it does is to load the data. It's basically the data, as I said, is the data that I digitize. It corresponds to these data points here. And then the second part loads the Antimony model with the parameters that we estimated to get the previous vital load curve. And we saw already that we get a dynamics of the cell transitions, how cells transition from one state to the other. We got values for the cytokines in the tissue and the lymph node, for the number of T cells in the lymph node and the tissue, the vital load curve, and then finally we can compare the data points to the data set. And I do have to say I'm sorry, because in this line that you guys see here is their model, and I forgot to mention that because I forgot to include that, so we could compare their model that they had originally with the one that I have here, that we've created here. It would make it easier to compare. But in general, we can see that our model is kind of fast just by looking at this. We can see that the model is fast at the beginning. The decay, and this comes, the decay is much shorter, and then the slow phase is much shorter, and the decay rate is much faster. It kind of looks very different when you put it in when you compare them when you overlay them. But yeah, so I mean, we're not terribly far off for considering that we were estimating these parameters. Okay, we will assume that it takes 2 days to the lymph node to the signal to reach the lymph node. Considering all those factors that we were basically guessing to an extent, we actually have a really good initial guess. Okay. But now that we have that, now that we compare that, we can actually use the optimization algorithm to try to get a better fit between our model and the experimental data. And just as a refresher, I included some of the slides from the previous, from the section where we did this already, right? And the idea is that we need to be able to fit the model to the data. We need to be able to quantify what is called the residuals. And the residuals is just the difference between your model and the data. It's the distance between your model prediction and the observed experimental data. And once we've quantified, once we counted that difference, right, the difference between the model and the data, then there is different options as to what we do with that distance. But the standard way of the standard way of quantifying the fit between the model and the data is by using what is called the chi-square function, where we measure those distance, we square them, and we basically add them. It's just a simpler. There's more complicated forms that can be used, but this one is the simplest one. Hilbert discusses a couple of different other functions that can also be used in his book. And then once we define, this chi-square defines what is called the objective function, right? What do we do with the distance, with the measurements of the distance between the model and the data? Then our optimization algorithm is gonna minimize that that objective function, which is another way of saying that it may try to minimize the distance between the model and the data. That optimization basically explores different combinations of the parameter values, measures the distance of the model and the data, which is represented here by this surface. So in this point, in these points, the distance between your model and the data is very big. In this point, the model is better, and this is the best solution to the model. It's the best fit between your model and the data. So what the fitting algorithm does is that it randomly is not the right term. It chooses certain points in this parameter space, and the algorithm is just a way of how do you decide what's the next point that you're gonna choose. Different algorithms implement different ways of exploring the space, and then tries to find that smallest point in this space, which corresponds to the smallest distance between your model and the data. So the first thing to be able to implement the optimization is that we need to define this objective function. I'm gonna walk you guys through. Last time I didn't spend time walking you through what the code is doing, but I'll spend a little bit of time here talking about it. So as I said, the cost function or the objective function takes as an input a different values for your parameters. So basically it takes a point in this space, which is a specific value for each of your parameters. So the first thing I need to do is take those values, load my antimony model, and then assign the values to each of the parameters of my model. And this is what these lines 3 to 6 are doing. So basically P is the set of the parameter values that is gonna be produced by the optimizer. I load the model, and then I basically create a dictionary that allows me to assign parameter values with the model parameters. Is that clear, or should I explain it again? So once we load the model and then we assign the parameter values, then we can simulate the model. And it's important that we simulate the model for long enough that we cover the data that we wanna compare it to, right? If I simulate it for short times, then it means I cannot compare it to my whole data set. So I need to at least simulate it for long enough, right, that I can compare it to my data. And in this case, it means simulated at least longer than 10 days. In our model, in this code, we're not asking the user to specify how long. We just take the last point in your data, and then we decide how much longer after that to simulate the model. Okay. And then once we've simulated the model, then we need to compare the model to the data. But the problem is that the way the model is simulated, the time points on the model might not correspond one-to-one to the time points in the experimental data. And there's multiple ways to solve that. I think this is my preferred way because it's simple, but it is an approximation. And I'm gonna explain what this one's doing. Maybe I can draw. Let me see. So let's say, okay, I'm gonna draw here. Let's say I have, that's a very well color, you guys can see. Let's say, no, that doesn't work. Okay, let's say I have that my model looks like this. It's kind of like an approximation of the viral loop curve, I don't know. That my experimental loop like this, my experimental data looks like this. The model, depending on how many points I simulate my model for, the model might produce points that might or might not match the same times that my experimental data was collected. So for example, none of this do, right? So let's say this one does, but this one is a little bit off. This one is clearly doesn't correspond. This one doesn't have a corresponding point, right? So what the algorithm is doing here in these lines is basically looking at the ones that are the closest. This one is the closest, this one is the closest, this one is the closest. And then it's comparing the model to it's comparing the data to the model point that is closest in time, even though if it's not exact. Of course, if I have very few points, then that comparison might be very bad. But we included enough points, right? So we had 10 points and we simulated the model for 100 points. We have 100 points in our model, in our simulation. So we included enough points that we ensure that they are relatively close, even though they are not exactly one-to-one at the same time. Okay, and then after that, then we measured the distance between them, right? Which is what we're doing here on line 12. And then we squared them and we add them. So that gives us diff, which is, and then we take the square root, which is the chi-square, right? Which is the function that I described earlier. So any questions about what this code is doing? Because we actually might ask you guys to play a little bit with this code for the homework. So I want to make sure that you guys understand, at least have a sense of what this is doing. Okay. So next, good. So next we display, and this is just for visualization purposes, we are displaying the model and the simulation with the parameter values passed on from the simulator, so we can see in real time what the model looks like given by, because you don't know what the parameter values are, the algorithm knows. But a way of visualizing what the algorithm is doing is to plot the model and the data and see how the model is closing the gap between the model, how the optimizer is closing the gap between the model and the data. So this is what this part is doing, this part here. I'm not gonna go into the details of this now. I'll probably come back to that later when we're gonna have a full class on parameter identification. Okay. So the next function that we define is this function. This was not included in the previous version of this code. And the optimizer, this is basically additional information that we pass into the optimizer. And what we're telling the optimizer here is that if the difference is small, the total difference between my model and the data is smaller than this value, then we're asking the model, the optimizer to stop. And the reason we're doing that is because the optimizer has its own rules to determine success. And those rules might or might not be the same rules that you as an user want. Maybe the optimizer is obsessed with minimizing like a particular difference, but for you, the fitting is good enough. So you can tell the optimizer, okay, below this, as a user, I think that the optimization is good enough, so you can stop there rather than continue going. And this is what we're doing here. And that value, we determine that specific value by playing with by running the simulation once and seeing what it was doing. Okay. So the next part of the code introduces all of the parameters that we're interested in fitting, which are basically all of the parameters of the model. I forgot to mention, and this is important too, and I'm sorry that I forgot this because this is really important. If you guys look at line 12, line 12 is telling me that I am comparing the results of the model for the variable V with the experimental data. But what would happen if I have two data sets that I have, let's say, a vital load data set, but I also have a count of immune cells in the tissue? What would you guys suggest would be? So without thinking the code, what do you think? What do you guys think is the way to do that fitting, like algorithmically rather than in the code? So the question is, in this particular example, we just have one data set, which is the vital load. But let's say you have vital load, but you also have number of immune cells in the lymph node, let's say. What do you do?
And we're still treating these as two separate parameters, right?
Right. I mean, the parameters are going to determine what the curves look like, right?
Oh, right.
But what you have is two different curves. Right. So if we look at back here, right, we're trying to fit this curve to the data, right?
Yes.
But in principle, we could also fit this one to the data as well. We could fit two instead of just one, right?
Right.
And then let's say, just as an example, but as a very simple example, then I would add another term to my chi-square equation, but instead of just comparing the TCID, I compare the immune cells to my, let's call it like EL, my number of let's say the data that I collected is the data of the immune cells in the lymph node. Then I can compare then my data to the results of the model. And might or might not be in logarithmic. It can be just normal, like absolute values. Natural values. Right?
Right.
So this one now, line 13 is saying I have an additional data set, and that data set is the number of immune cells in my lymph node. and then I can compare my data set to my model, and then I would add them. I would add that to the chi-square. And you might want to weigh them. You might want to say, for me, it's more important to fit the vital load curve than the immune cell, but both of them are important. But let's say the vital load curve is twice as important for me. So I can add a weight here, times weight, that says, okay, this data set is more important than my other data set. So I care more about weight, our fitting the vital load than the immune cells. But I am trying to fit both. Does that make sense?
And that makes sense now. I thought like we were trying to combine the two variables somehow, and I got confused for a second.
Right, no, but yeah, so what you want to do is, hold on. What you want to do is compare the data, and then you might want to weigh them or not. There is also, you also have the chance, and we'll see this, we can play with this a little bit later. You can weigh the points differently. So in this, with this equation, we're weighting every point in our data set equally. We're treating every point in our data set as equally important. But you might want to say, okay, these last 2 points are not as important as the first few points. So I can assign a different weight to my data points. And you can see that actually in their paper, they did not try to fit those 2 points at all. Like, I mean, yeah, they might have, but it's clear. I mean, if they might have, I would expect the line to go somewhere in between those points rather than, yeah, I think they didn't, right, James? Right. It's fine. You're muted, but it's fine. Yeah, I think you can tell by the fit between the model and the data that they basically ignored those 2 points at the end. And that is something that we can do, and we can play with it. Later on. Let's show how the code, and then we can play a little bit with it. So let me. But I did wanted to show you guys how simple adding another data set would be in the model, in the objective function that we define it. So once we've specified the parameters that we want to fit, then we assign the original values of these parameters, and these are the ones that we got from we estimated already. And then we define bounds for the parameters. And this is important because that determines let me go back here. That determines the limits of this space. You don't want that space to be infinitely large, especially with certain types of optimizers, because it would take forever, like literally forever, for the optimizer to finish that. So you want to specify certain bounds. So this is the bounds that I'm allowing the optimizer to explore. Here what we did is, okay, explore the space that is such that the parameter is 10 times smaller and 10 times bigger than the ones that we originally estimated. And the reason we did that is because we already had a pretty good guess at the beginning. So we knew that the parameter values had to be, we were close. We were not there, but we were close. If your original guess is really bad, then you might need to have a bigger space. You might have to constrain your optimizer less and allow it to explore a bigger parameter space. Yeah, and so once we run the cost function with the original parameter values, the original values of the parameter sets, we get this comparison between our initial guess and the model.
How do we know if the parameters that we chose are way off? Like, is that up to us to come up with some kind of metric for it?
So it's kind of like a philosophical question rather than a scientific question. James, you can jump in because I know that topic you.
Well, way off is a matter of convenience. You'll find that if you start with parameters that are way off, one of two things happens. Either the optimizer will happily converge to some result that doesn't agree at all with what you're trying to fit, in which case you're stuck in what's called the local minimum of the optimization space, or the optimizer never converges at all, in which case it tries and tries and tries and fails to give you a result. And so fundamentally, starting with reasonable guesses means you start close enough to the solution you want that your optimizer can get you there. In general, in these high-dimensional nonlinear systems, there is no perfect optimization. And if you start too far away in parameter space from the final result, you won't be able to get, the optimizer will either fail, give you an incorrect result, or will continue to try forever and never give you a result. And so, in fact, I was just, while Josh was working on this, I was just playing. We had talked. There's an issue with you'll get a result when you run this, and there's an issue with that result. And I was trying to put into the optimizer the constraint to fix that problem. So that was what I was doing while Josh was talking just now. And when you do that, it's not so trivial to make it work. But optimization is a bit of an art. When you do it a lot of times, you get a sense of which parameters are most important. You can do some mathematics, but it's called sensitivity analysis, to try to identify which parameters are the most important. One thing you could do is freeze all but one or two parameters and optimize only on one or two at a time. And optimizers tend to work better, and you can then go through that sequentially. But there is no universally good way of optimizing. You can imagine it this way. Suppose that your perfect solution is like the cup on a golf course for hole number 18. And what you want to do is get the ball, which is the parameter values, into that cup, which is the optimum. If you try to roll the golf ball around on the golf course, if you don't start on the putting green, there's no way that randomly rolling the ball around is ever going to get your ball into the hole. And so optimizers are a little bit like that, where you're trying to find a small hole in a very rough landscape. So, I don't know, Josh, if that's what you wanted, but that was what my comment on it.
Right. Yeah. And again, I think that is a, okay, I do not want to say that it's all an art, because part of the stuff that we spent the last 3 weeks doing was constructing the model such that we had an initial good guess, right? So when we were choosing the parameters, when we were constructing the model and looking at the curve and making sure that, okay, the first part, we replicate the first part of the curve, we replicate now the saturation level, now we replicate the slow decay, we replicate now the fast clearance. We constructed the model purposely such that we had an original good guess. And that is stuff that if you understand how the different scales of the model work, you can definitely do that. And that's something I'm still learning a lot from James, because you can determine, okay, this is what this parameter is going to control this part of the curve. This parameter is going to control this part of the curve. And as you get more familiarity with how the dynamical systems work, then you have a sense of how the parameters are controlling the different parts of the curve and what an initial good guess of your model is. Does that make sense?
Yeah.
Okay, cool. So So the next part, what we do is that we actually run the optimizer. And if you guys look at the notebook that I uploaded, it's already found a solution, but you can look at the history of what it did. We started with our initial guess, and it started moving and getting closer and closer to the experimental data. And you guys can see that in our model, we are definitely fitting for those two last points, which is different from that model, because in that model, the model is clearly not intended to fit those 2 points. But in our model, it is. So you guys can see how it's trying to fit all of the points. And even though it has fit all of it at the beginning, it's still trying to work a better fit for the tail of the model. And then the reason we stopped when I was talking about that callable iteration, callable function, is because if you guys see, it's been all of these iterations, it's been pretty much, this number here is the residual function, or the value of the objective function. And when you look at it, it's clearly not decreasing by a lot. It's decreasing by 4 or 5 decimal points. So that is a level of accuracy that I think that for the purpose of this class and the exercise, it's good. It's trying, like, it's not changing a lot. So that's why we included that condition asking the optimizer to stop, because it had reached already a good enough fit. And then the wrapper, which is called LMFit, it's gonna report the values of the parameters and your initial guesses. And let's compare them. Let's see. Our beta value was much smaller, at least an order of magnitude smaller. The decay of the virus was small.
Beta is only off by a factor of 2.
Oh, right. It's a factor of. It's just I was looking at the exponent, but it's really just a factor of 2, right? So we were not that off. And it compensates because the virus now decays faster. So the virus is more infectious, but it decays faster too. But all the other parameters are pretty much like in this. We were already quite close, right? So it optimized it for sure, and we have a fit, but we were already quite close to the actual results. And then now we can come back again, and then now look at how all of the other variables of the model behave when we fit the data and compare it to what they looked before for the previous model, for the estimated model. And then let's see a couple of them. In terms of the cell population, it seems like cells, you have less cells with this new model. You have more cells in the eclipse phase than you had in your in the original model. And also cells spend much time, a little bit longer in the productive phase before they start dying. They die much slower. And that's why this curve is also dented down, because the cell death is happening much slower. But they basically reach this maximum around the same times for each of the values of the curves. Some of the values changed, but right. But it's not terribly different, except that you have that. What is happening in the model is that stuff is dying much slower than in the original model. In terms of the cytokines, they look pretty similar to me. There is a little bit of a difference here, but I don't think it's very important. But I think in terms of the T cell, it's really important because the order is really huge. So how many cells? No, it's not. Wait, I thought. No, we were actually quite close. It's just because the axes were shifted. But yeah, we're kind of close. So we were not that off original to start with. We did, and most of the work of the model was achieved. The fitting was achieved by letting cells live a little bit longer, adjusting the parameters so cells live a little bit longer. And with that, you can sort of like get a curve that fits better the data. But in terms of the because my concern was that if there was a difference between T cells in the lymph node and the tissue, that might be an indicator that your model is not. If you were fitting that to the data, then it might not work. But the fact that they produce the same, it's kind of a good indicator that the model is constraining that. James, but you wanted to say something about what this model is doing.
So I guess something to think about is if you look at, so we fit the viral load curve, and that worked. And I think, Josh, probably you want people to run this, so you want people to actually go through it.
Let me throw a poll, so I make sure that everyone can at least run their version of the code.
It'll take a little time for the fit to work. It'll take 4 or 5 minutes for the fit to run. And what I was going to suggest, and I don't know exactly what you've got planned for class, but maybe it would be worth trying to make sure that people understand how to manipulate the code, even if it doesn't exactly do, even if you don't know all the details. And you could do this with them perhaps, would be the following. Let's look at we fit with the viral load curve, but if we look at the red line here, here, we notice that all of our cells are dying. If all the cells in your lung died, you'd die long before all of the cells died. You'd die when a quarter of the cells had died, or even something like that. You develop severe pneumonia when you have 10% of the cells in your lung die. You develop irrecoverable pneumonia when you're on the order of 20% of the cells in your lung die. And so this is fit the viral load data, but these animals didn't die there because you were making measurements. So by definition, they weren't dead. So at the end, we have all the cells dead. And that makes sense because when you have all the cells die, you stop producing virus. That makes sense. But in fact, we know in reality, when you get flu, you can get sick, you may get some pneumonia, but you're not going to have every cell in your lungs die before your immune system clears the virus. And so you could ask the question, if we say that we want the final fraction of cells that were infected or uninfected to stay high, suppose we want half of our cells to remain uninfected at the end, what happens? And so I think a worthwhile exercise to do together would be, and Josh, you could do this on the fly perhaps, would be to take the code that you have, find the value of T at the end of the simulation, divide by T0, which is the fraction of cells surviving at the end, which is a number between 0 and 1.
Right.
take the difference between that and some target like 0.6 or 0.8,
Right.
square it, and add that to the differential.
Yeah, I need to take 5 minutes, but we can while people load their codes, because we can work on this together. Then I'll run to the restroom and come back in 5 minutes.
Okay, so during this time while he's off, why don't you try making sure that you could load and run the models as they exist? Watch the outputs of the simulation as it's going through. So watch the individual replicas. You'll see that the residual, that number at the top, sometimes goes down and sometimes goes up. The fitting algorithm doesn't always go to better solutions. It tries to get better, and sometimes it'll jump past a good solution, or it'll get stuck and it'll back up and try something else. And so you'll see that initially it explores the space of parameters rather broadly. Then it decides on what it thinks is a pretty good solution, and then it'll try to get finer and finer in terms of its evaluation. But you should be able to see that. And so it's worth scrolling through those outputs as they're happening and watching those. So why don't we do that while Josh is away? It's also, if you need to get a cup of coffee or run to the toilet or something like that, you could set that running, because it'll take a few minutes, and then go and come back. But I think it's important that everybody try running that together and see how Optimizer works. NanoHub will limit the amount of computer time you use, and so it's possible that if the Optimizer really takes a lot of time, NanoHub will time you out. But I don't think that's going to happen here. At least when I'm running it, it doesn't happen. So why don't people we'll take a 5-minute break, say come back at 6:45, and we can see what happened. And why don't people be ready to report? Did they get the same values that Josh got, that he's got here on the screen? Did you get the same time series that he got? I think that's a pretty interesting thing. Maybe you get a different result. One thing if you haven't discovered it yet, that you may want to do is in your when you're running the simulation under kernel, if you pull down kernel, there's something called interrupt.
Right.
And so if you get tired of watching the optimizer grind along and you want to do something else, you can hit interrupt and stop it.
Right. But we can, if it's taking too long, for example, we can increase the residual that we want, that we're gonna, let's look at, okay, let's look for a second. What is an acceptable residual here? Which is a little bit of what I was talking before. So you see that at 53, it's already spent a lot of time at 53. Even 55, below 55 is good. Yeah, we can set it to 50, below 55. We will save a lot of this computation. Okay. So let's go back. So what James was discussing before I had to leave was that when you look at the results of this model, even though we fit the vital load curve, when we look at the slope population in this, for the particular parameter values that we chose, all of the cells are dead at the end of the simulation. And we want to explore whether the fitting algorithm can find a set of parameter values for which there is some cells that survive at the end of the simulation. And that would correspond to a scenario where the immune system kicked in and controlled the infection before the infection spread throughout the tissue or throughout the domain that we were simulating. I'm thinking spatially. So the first thing I'm gonna do, just as an example, is that instead of trying to, as I was explaining, we were trying to fit the model to all of the data points. But when we look at the original data, it seems like they did not weight those two last points the same way they weighted the previous points. So what I'll do is I'm gonna just try to not fit those two last 2 points. So the way I do that is so here in line 10, I go over the times that I want in the experimental data that I want to fit to the model. So if I, let me rerun this. Okay, so I'm gonna. So what I really want to do is to ignore those two last points at the end. And probably no one who knows better like the syntax here, but I think it's something like this. I'm gonna try to guess. Yeah. Okay, I do know the syntax. So basically, I ignore the last 2 points. I cut my data. I slice my data, so I ignore the last 2 points in my experimental data. So I know that this is a syntax to do it. So I mean, I already cut it, so I could just try to fit the model to the data, but let's do something. Okay. So when I come to my function, in my function I go from in line 10, I'm basically iterating through each point of my of my data set. So what I'm gonna do is just I'm gonna -2 here, and that's gonna what I'll do is that what that will do is that it will try to fit the data. It will ignore the last 2 points in my data set. Right. But then and then now I can run the optimizer again and look and see what it's doing. Basically, I told the optimizer to ignore those last 2 points in the data set. It's running. It's going to take a little bit of time. So now it's spending some time trying to, because it's ignoring these 2 points, it's trying to fit this and then this exponential decay, fast exponential decay there. Okay, let's see how it's doing. Right. So you guys can see there that it's ignoring those two points. And actually, when we compare that to this, the one that they're reporting in the paper, it looks much closer to what they had in the data, what they published in the paper. So that was something I actually wanted to assign you as a homework, but it was not extremely complicated. I just basically excluded the last 2 points of my data set in my fitting algorithm. And I think it found a solution. Yep. And then now here's the new model with the solutions, with the new solution, but I ignored those 2 points. Okay. And that was just like a one-line modification. Now, what we want to do is that we're fitting just the vital load curve, but we also want to add an additional constraint in our fitting. And the constraint is that we want the number of the fraction of infected cells. We want some cells to survive at the end, right? At the end of the simulation. So what we are going to do is any ideas of how we can do this? Just conceptually, rather than don't worry about the code. I want to think about it conceptually. How should I do it conceptually? What should I compare to what?
Can you repeat the question?
Right. So so we fitted the vital load, but now we want to add an additional extra constraint in our algorithm, and the constraint is that at the end of the simulation we have to have some cells survive. So we want some cells not to be dead at the end of our simulation.
And this, we would want to put this in after we do the fitting.
We want to do it while we're fitting, because we want what we basically want. We want to ask the fitting algorithm to do two things: fit the vital load curve, such but that at the end of the simulation I still have some surviving cells, right? So I'm basically comparing my vital load curve to the data, and then there is something that I need to do with this red line, which is the number of dead cells.
Put a maximum value on it.
Right.
Maybe like once if we hit it, we stop.
Right. Or we can ask the model, try to do your best at fitting these two things, these two conditions, because there might not be a solution. Maybe James found one, but I don't know. We don't know in advance whether the model can find within the parameter range that we set, whether the algorithm can find a solution or not. But the idea is that we would add, let's say, to our equation, we would add. Okay, first, let's start with how many cells I have at the beginning of the simulation. That it's called T0. That's the number of cells at the beginning of the simulation. The number of dead cells at the end of the simulation is going to be this one. And I had to include it as a variable that I want to follow in the model. Right? And then and then the fraction of those is, right? So what I mean when I say I have some cells to survive at the end of the simulation is that this number is less than 1. Right?
Yes.
So that's what I want, right? So what I should do is that if at the end of the simulation this number is 1, or what fraction, let's call it like 90. So we want 90% of our 0.9, I'm sorry. We want at least 90% of our cells to survive at the end of the simulation. That threshold we can set up to whatever we want. And depending on that threshold, the model might or might not be able to find a solution. So if this is smaller than, if bigger than 90, then I add to my objective function, then I add some value.
I guess a simple thing to do would be just to say m dot d over m dot t0 minus 0.9 quantity squared, and add that to your difference.
Right.
Well,
but then you want that exactly. Okay, that's
D is dead cells. So you actually want it to be 0.1. You only want, say, 20% of the cells to die.
right.
So you could say
It's
m dot d minus over m dot t0 minus 0.2 quantity squared, and add that to your difference.
Yeah, you're trying to do something a little bit different, right? What James is, you're actually asking the simulator, the optimizer, to find a specific number, whereas I was asking whatever number, but that I at least get this amount, which is, I mean, it's two different questions, but yes. So right. So basically, what I'm gonna do now is I'm gonna ask the optimizer to find a solution such that at the end of the simulation, the number of surviving cells in this case is 90%. So 90% of my cells survive at the end of the simulation. That's a lot. We'll see if that works. So before I move on, I want everyone to type this and ask questions as to whether it's clear what I'm trying to do. I'm gonna create a poll to ask everyone to add this. It's basically add this to the code, but just make sure that you understand what is it that you're adding. And if you have questions in terms of what is it that this is intending to do, yeah, please do ask.
Sorry, Josh, in line 13, do you want that to be greater than 0.1 or do you want it to be minus 0.1?
Oh, I'm sorry, no, minus 0.1. So we want it to be, right, 90% of our cells to survive. Do you find a solution for that one? I'm kind of like curious as if that would work.
I'm running the simulation with the less than or the greater than 0.1 right now. I'll let you know if it spits something out.
Okay, cool. I mean, that might work as well. I mean, it's like, yeah, as I mentioned, one of them is you're asking the model to give you exactly that, and the other one you're asking to give the optimizer any that it produces more than 10%. And then so let's do something, because since there's a couple of people, we can test different hypotheses here. So I'm gonna run it for 10, but can someone run it like for 0.5? So half of the cells survive and half of the cells don't. And then we can then compare results at the end. I'm sorry?
I say I can run it at 0.5 instead of 0.1.
Good. Yeah. So I'm gonna run that one and see.
I'm having an issue, which is that the final fraction of cells that are dead seems to be bigger than 1 when I do it, which shouldn't be possible.
Yeah, that's kind of awkward because we're not getting any supply of cells from anywhere.
It might be worth plotting the number of dead cells and
Right.
the number of uninfected cells inside the loop so you can see what it's doing.
So we can do that as well.
In case it's doing something strange, so I'm trying to debug.
Yep. And then what you want to show is what I'm sorry, James, what do you want it to show? The number of infected cells?
I would plot T and D versus time.
Right.
So that you could see whether it makes sense. I mean, you could plot I1 and I2, but that you don't have to think it's T and D or not.
Well, and wait, if I divide it, it's fine. Okay, let's see.
I guess you could rescale by T0 if you like. But
right? But it's fine because it's, and again, you don't get more than cells that you. Right. The problem now. So this is another problem. So the problem now is that when the values that we passed on to the callable function to stop the optimizer were based on the previous metrics that we were using, and that one is now. And you guys can see that now the model is doing quite well at 1.04, but it keeps going. So in this particular case, I could modify it to make it shorter, to make this number bigger. The residual that is acceptable to be is 1.04. Is it, or yeah. So, Antonio, how is yours going?
Mine was still spinning. I interrupted it, and I'm gonna set that. I'm gonna reset that value because I still had it set at the 0.55.
Right, but then yours might depend as well on what I mean what the value is that it settled in.
It looks like it was settling into like 1 point. The latest one, 1.0377.
Yeah, you can put it on 1.304, and I think that's gonna be fine. I'm gonna actually.
Yes, the one thing, Josh, is it's not plotting it's not actually showing the time series for T and D.
Mine, because mine was I'll that I modified later. So
So, because I can't tell if it's actually doing anything or not.
wait. What's wrong? Oh, that I'm not recording T. So Callie, what about yours?
My optimizer is still running.
Okay. So you might, if you're noticing that it's also settling into some value, you might actually want to stop it and then rerun it. Now, James, you can see that at least here is doing both.
I mean, this may also be one of the cases you were talking about where the parameters are so far from a solution where not all the cells die that it can't find it. So it's possible that the optimizer simply can't get to a solution.
Right. Which is yeah. And you might want to force it by increasing, putting much more weight into the fraction part instead of the vital load part. Let's see. And again, some of the, again, that's why I wanted people to try different different criteria to see whether, because we don't know in advance whether there will be a solution or not. Yeah, I think at least on my end is not, it's not putting a lot of, it's not putting a lot of effort into fitting that latest part.
I think what's happened is that you're so far, basically, all the cells die long before you get to the end.
Right.
And so changing parameter values from this initial condition doesn't change the fact that all the cells die. And therefore, it has no way of finding out what to do. So you'd have to, by hand, find a situation where not all the cells die. And we know how to do that. We know how to evaluate R naught for the system. R naught would be the rate of viral production times beta, the rate of infection, divided by the lifetime of the cells and the virus. And so we know that if we make beta smaller and D bigger, that would reduce the fraction of cells that die. So probably one has to do some hand fitting before one does this. So I don't know, maybe I sent you on a wild goose chase, Josh. I'm sorry.
No,
I mean, we talked about doing this, but.
Yeah. But what I'm a bit concerned is that we might not have time to replicate the papers.
So maybe the thing to do then would be to start reviewing the papers. We'll wind up doing, we only have 40 minutes. So why don't we start reviewing the papers, discuss the replication, and then we can do the full replication next week.
Right, and at least have people implement the models and at least execute them before class, so we don't have to spend time on that part, and we can concentrate on the results. But I want to hear from Antonio and Kali to see whatever they got.
Mine aired out, so I had to reset the kernel and rerun everything.
Okay, yeah, that sometimes happens as well.
I also interrupted the kernel and restarted.
Okay.
But my model was definitely converging. It was close, but it just kept going on and on and on.
Can you show the line where you did the plotting? I just want to make sure I'm doing exactly what you did.
Okay. So, James, were you able to?
I got what I needed from your plot. But I'm poking around to see if I can find the parameters. There's some parameters that work.
Right. Did you find the
Right.
notebook where you play with the? Do you find that notebook?
Let me take a look while you're covering the other things.
Okay. Okay. So the last couple of minutes of class, well, what we were intending to do for class today was to replicate some papers, but so we'll let's look. We probably won't have time to implement the papers, but we can start discussing what the papers are doing, and then next week we can spend some time trying to replicate some of the results. Okay. So the first paper that I shared was I'm sorry. It's called an in-house model of HIV incorporating latent infection and viral mutation. I wanted to say that these papers are a little bit heavy on the math, but the math is not necessarily super important to reproduce the results of the paper. They are basically proving certain mathematical stuff about the dynamical system and then showing what happens in the simulations. So in this model, what you have is basically you have two type of viruses. So let's just draw the flow diagram of this, or the arrow diagram of this model. So in this model, you have some, you basically have the you have the standard model where target cells become infected, and then these infected cells let's see what it's well, latently infected, and then because in HIV these cells in HIV the infection model works a little bit differently. So cells go from infected, right, to this early phase of infection, and then some of them also come into a latent phase, and how many of them go to one versus the other is determined by this fraction. So P goes to latent, 1 minus P goes to infection. Latent cells can become infectious, and they die, and the infected cells die as well. So that's basically the model with respects to one virus, right? And then these infected cells produce virus. The virus dies, and then the virus comes here and infect the cells. Okay, that is one side of the model. So basically, you have a virus. You have the virus. The virus infects the target cells. Some of the infected cells go into this latent phase, but some of the cells become actively infectious. And then the latent cells spend some time in the latent period, but they can also become infectious, and then they produce virus, and then the cycle repeats. But in this particular model, they have a two virus competition. So they have a wild type virus that they're calling S, and a mutant type virus that they're calling VR. I'm going to write here. And what happens is that they behave equally. So cells can become infected by either the resistant or the mutant virus. So this is basically the arrow diagram of the whole system. You have two types of viruses, and then they can infect the same target cells. The cells become, the type the cells become depend on the virus that infected them. And then there is a couple of parameters that control the rate at which this infection happens and how cells move between the different phases, how fast they die. This is alpha R, and this is alpha S. So there's a bunch of parameters that control all of this. In terms of the, let's see what they do in the model. So let's see what they do in terms of the results. So they estimated these parameter values, the values of all of the parameters. I haven't read the full paper to know how they estimated them, but since they have references, that when they have references, that usually means that they looked at the values of these parameters from additional sources, which might or might not be a good strategy depending on what you want to do. And then they simulated the model with those parameter values. So this is something that you guys can definitely work on as a homework before the next replication, before we replicate the full results of the paper. Because basically what they've done here is they implemented the model, they assigned the parameters as in the table, and then And then here, what you're looking at is the vital loads. Yeah, these are the vital loads of one strain of virus, and then the resistant strain, and then basically what they're plotting is the number of healthy cells. You're gonna have all of the other variables, but if you just want to look at this 4 plot, yeah, this is just the number of healthy or uninfected cells. Okay, so that's something I'm gonna assign that as a homework. You guys have to reproduce, because this is quite straightforward, and this is something you guys can work on by yourselves. The next part, sorry, I'm trying to delete all the drawings, but the bar won't show up. Okay. So, we did spend some time here, and this is what we're gonna spend some time on when replicating this paper. So what they do here basically is that they take 2 parameter values, and then it's basically R naught. They change the R naught for both of the of the viruses, and then they identified different regions in which one virus dominates as opposed to the other, or where infection fails to happen. And what we'll do next week is simulating all of these space. It can be complicated because you will have to change. It's a multi-dimensional change. So you change R1, R naught for one virus, and then you change a lot of R naughts for the other, and then you combine this, let's say, pairwise change in parameter values. But instead of doing that, we can just draw a line here and a line here, and simulate the model by performing a parameter scan and seeing what the steady state is, what the simulation looks like at the end when we look specifically at one line here and one line here. And we'll do that next week. That's part of the replication that we'll see. Right, and then this is similar. It will look something similar to what the results that we will get will look something similar to these plots, and then we can compare them to the results that they're getting here. The stability analysis, I'm not sure we're interested in doing, but then after we do that, then we can explore some of the drug therapies that they test in the paper by changing. There is a specific parameter that I did not talk about, but that represents drug therapy by decreasing some of the parameters that control how much virus is produced, or the infectivity of the virus, or how much time each cell spends in the latent phases for each of the viruses. So we will spend some time doing this as well by looking at how adding drug therapies changes what happens at the end of the simulation for the different parameter values. So this is doable, but again, what I would like you guys to do as a homework is to replicate this figure 1 in this paper. So you will need to write down the antimony model of this, given in these equations, plugging these parameter values, and then try to replicate, make sure that we can replicate this curve.
Joshua, I have my results from the optimizer. If
Okay.
you wanted to go back to that.
Yeah, and what did you get? Do you want to share?
Sure.
Okay.
So the final fit looked like this.
Right.
Here are our variables.
Mm-hmm.
And then I found that all the cells still died anyway.
Right. That's what James was talking about. Yeah. James, can you explain again what's happening here? James, are you there?
Yeah, I'm here. Sorry. So this is what I was saying about optimizers. We're starting from a situation in which all the cells die, and anywhere nearby in parameter space, all the cells still die.
Right.
So the optimizer has no way of knowing what direction to move in parameter space to make the cells not die. And so it gets stuck. It gives up.
Okay.
So what I was trying to do was to say, okay, we know that if I make the virus less infectious, fewer cells should die. If I make the amount of virus produced by each cell when it's in the I2 phase, fewer cells should die. If I make the lifetime of the infected cells shorter, fewer cells should die. If I make the number of immune cells, the killing efficiency of the immune cells higher, fewer cells should die. And so I forced my initial condition into something where the fraction of the cells that die is smaller, which I could do. You don't, if you scroll up in your before the one before the fit, if you keep, so in this box, number 6, if you multiply and divide these parameter values and then re-execute this, it'll give you a plot of the viral load versus time, and then also the number of uninfected dead cells. And so by hand, quickly in this number 6, you can play around with the parameters. So for example, if you make the killing efficiency, which is DEI2, if you multiply that by 5, for example, you could run that again and see what happens. Just click execute on just that box. Right. And we can try that and see what happens in that if we look down at the plots underneath. So you see here, the virus gets cut off much faster. It would be helpful for doing this if you add those 2 plots that Josh added to his code. So that if you scroll up to box 5, here where you do the plot, if you copy this plot, sorry.
I'm gonna paste what you need to put in there.
Yeah, if you copy this plot line here twice, and then another plot show after the plot show, and you can plot here. You're not evaluating. Oh, okay. So in your run, you're not evaluating D. So one thing here is that you're not in your output for your s.simulate, you don't have D and T. So it doesn't know what the value of s.d is at the end. I don't think. Maybe it does. Let's see. Maybe it's all right. Maybe m.d is all right. But I would But if you're going to plot it, you certainly need to put them in here. In the S equals S dot simulate. You need to add T here and S. And then you can duplicate this line and make it S, plot S as well as T.
As well as T.
I'd plot, just copy this line and paste it below. And then replace T by S. I'm sorry, D. I said S. I meant D.
Okay.
There.
I was going to say that made more sense.
Sorry. I see S, and so I said S. And then you'll have to move these two lines below this show here. And do another show. Okay, now execute this cell. And then that subsequent cell here, this one. And now you have that. So now with that, you can see what's happened here. The virus disappears much faster than it did before, but still all the cells are dying.
Right.
So we could try, if you make the beta value smaller here, so that we could divide that by 10 to start with.
Okay, so now.
It's still everybody's still dying.
Right.
So we have to think about what we could do to make it. Reducing beta should make fewer cells die. Increasing the death rate of I2 cells. That's another thing we could do. So that the D parameter for up here would go up again. D here, we make that bigger. Then the cells are infectious for less time, so they should cause fewer cells to become infected. So you could multiply that by, again, I'd try 10 until you get something that works. Multiply by 10 then. And so again, I'd have to play around with it a little bit, but I had a parameter set that at least gave me not all the cells dying, and let's see what I had to do to get that. I divided beta by 20. I divided P by 10. I multiplied D by 2. I multiplied KE by 2. This one, the next one. That KE. And I multiplied DEI 2 by 10. So. And that's it. That's all. Oh, that's all there. Okay, so that works. So now at least you have an initial condition where not everybody dies. Of course, the agreement with the viral load is terrible. And so then the question is, starting with this, could you keep the not everybody dying and get the viral load to agree? You could come up with some other values of parameters that would probably also not have everybody die. But at least you have one set of parameters where everybody doesn't die. And when I run this, At least so far, it hasn't been able to. It hasn't been able to find the solution. It's not terrible, but it's not able to find something that works. My guess is that there actually should be a solution. In other words, I think there is one, but we're probably starting too far away from it. But I guess you're getting a lesson in the fact that parameter fitting is more of a art than a science, maybe in a harder way than we intended.
Yeah.
But in my case, yeah, it's not. I mean, so if you look at what you've got, so here in your case, it's given up on trying to keep the cells from dying, right?
Mm-hmm.
It's gone back to a situation where all the cells die.
Yeah.
And if you let that keep running, I believe it will recover your original values. So it will basically abandon the attempt to keep the cells from dying, and eventually it will refit the viral load curve properly. It may take a while, but at least that was my experience. It was able to find again the viral load curve that we started with. And so then the next thing I did was go back in that weight function in the cell where you had defined the go up 2 cells. In here, where you added the differential to here, if you multiply this by a weight, like 5, then this term becomes more important than the other one. Actually, hold on a second. Let me look at what you've got here. So you say, oh, so here you're not actually looking at the, okay, not pi square root of diff. So this is a little bit of an unusual way to do this. I would probably put this outside after the square root because you're adding, you're taking the square root of 4. Okay, put that outside. So after there. And then and then there's some weight, as Josh said, you might want to adjust the weight. So you might want to multiply this outside of that square root by a factor. You should probably look at the numbers, but probably I've got it multiplied by 5. So outside of the whole squared item. So outside of all the parentheses. You have an error here, which you've calculated. You have another error you're adding to it. The question is, how much of each error do you use? If you multiplied by this by 0.1, then it cares a lot about the viral load curve and very little about the final value of D. If you multiply this by 10, it cares only a little bit about the viral load curve, and it cares a lot about the final value of D. So by multiplying that by, say, 5, you're saying I care more about the final value of D than I do about the viral load curve. And then when you rerun this, so you have to run this one and then this one again. When you rerun this one and this one now, and now when you rerun the weight, you'll find that it'll keep the value, the final number of dead cells lower, but it won't fit the viral load curve very well.
Should I
So
try that?
Why don't you scroll all the way down and see what it came up with, as far as it got. See, it's refit your viral load curve just fine. So even though you started very far away, it's recovered. Be interesting to see if the parameters came up with were the same. But you see it's refit the viral load fine.
Right.
It's come back to where you started, even though we knocked it off pretty far. But now when you give it a weighting of 5 for the other one, you rerun this, you'll see that you'll get something quite different. And the question is, if you play with that weighting function, can you find something that does both? You've seen that it can come up with a solution where not everybody dies, and it can come up with a solution where the viral load fits. The question is, can it do both at once? And I actually don't know the answer to that. Sorry, Josh, I hijacked your class.
Oh no, it's fine. That's totally fine. I think we we don't. I think this is important, and I much rather spend time doing this than trying to squeeze replication in 20 minutes. So I think this is useful, and it also showed how you can modify this code to add other stuff that you might be interested in fitting as well.
So one thing you might want to do, Josh, would be to write a little bit of a comment section or an introductory section, text section explaining how to modify this code to do the things that we were talking about today.
Right. I can absolutely do that.
Yeah. Okay. I hope people followed at least some of that. I think, Kali, I think now you've got something you can play with and see whether you can get it to work. And as I say, I don't know, because the model wasn't designed to do this. I don't know if it'll work, but at least it gives you a sense. And there really is no perfect. Well, the one thing that I could say is when you have these kinds of differential measurements, you're going to combine a diff for the virus load and a diff for the final value and a diff for something else, and you're going to weight them. You want to look initially at what the typical value of the first diff is, the second one, and the third one. And if you're going to have them solved simultaneously, then ultimately in the region you care about, those diffs all have to have roughly the same value.
Right.
And so you have to use your weighting to adjust that so that they're all approximately the same.
Yep.
Even with the weight of 5 added, it still looks like it's going to converge to a model where all the cells die, just from what I'm seeing right now.
That's possible. The way you wrote your final value calculation was a little different from mine, and so it's possible that the weighting needs to be slightly different. But I think you used, if you reshare for just 1 second more. If you can reshare your notebook on the screen, and let's look at how you define the error function. So you have, okay, so you used Josh's option, which was greater than 0.5. I used minus 0.5. Instead of greater than. So I tried to force it specifically to the value of 1/2 instead of and that was the difference in my result. So I think it might be worth trying that and seeing if that makes a difference. Yeah, that looks pretty close to what I had. And it wasn't able to do it. Now, it's possible that if we targeted 0.3 or 0.8 or something else for the final amount, it would work. Or it's possible that in your case, it'll jump to the original solution. But this kind of optimization is always a problem. When you want to try to optimize for multiple things simultaneously, you face the problem that there may not be any solution that gives you something that's good for both.
James, and I wondered, like, something we haven't done because we sort of like assume that all of the data sets are within the same similar ranges. You probably need to normalize the difference by the number, by the value of the point, by the value of the experimental point, or by the model, depending.
Well, that's true too. Although the values of the experimental point, yeah, well, you're doing it on the log, right?
Right.
So that does that.
Because then you're effectively weighting the points that are higher. Yeah, you're not normalizing by that.
No, but if you take the logarithm before you take the difference, then you're
Right.
basically doing that.
Right. No, but what I mean is right. Like but then when you compare number of cells to virus, right? Virus can be in the orders of magnitude different. So the difference then there might be.
No, but the residual you're getting is typically 1.
Right. Yeah.
For the particular way you calculated it. So the chi-squared for the virus fit is on the order of a few, 1, 2, 3.
Right.
So that means adding a number that's on the order of 1 is okay. So yeah, that's about what I got, which is it's able to keep the number of dead cells limited. And it's not doing a totally catastrophic job of matching the virus. It's getting them, I mean, it's going up, it gets the maximum right, and then it goes down about the right time, but it's not able to do fit the shape of the viral load. And it's probably premature to say that it's unable to do it because this model structure is wrong. Well, in fact, the model structure is wrong. And so if you made a guess that the problem now is that it's not able to do this because the model is wrong, you'd actually be right about it. In reality, you have to do a lot of parameter exploration before you make that, you're legitimately able to make that decision. Okay, well, I guess, Josh, you want to ask for questions, and then probably we've run over time a little bit.
Yeah, I'm gonna send as a homework. I'm gonna ask people to implement the 2 models that we were intended to replicate, at least to run the time courses. Both of them are shown in the papers, so that you can corroborate against the paper. And then next class, we can spend some time looking at their results and then trying to get meaningful results out of those models by doing parameter scans and other stuff.
And I guess the other thing is if people who are can give, you don't have to have slides, but maybe the people who were still getting started on their projects, I'd like everybody next week to briefly report on their progress on their projects. And if you're stuck, some people have come to talk to me. I'd like to try to meet with people as much as possible, either in the next 4 days or 5 days, to help people make progress on the projects. Because I know some people are having some issues getting started, so I'd like to work with you on that. Okay, anything else you want to discuss tonight? I hope that was educational, at least at some level. Fitting is a hard problem. Going through the code that Josh wrote is also something that maybe takes a little work. So maybe that's better to do offline, to walk through it line by line and see what it's doing. And certainly feel free to ask me or Josh questions about it. And we'll use that going forward also. Okay, Josh, back to you.
Right. So again, as I said, I'm gonna assign those as homeworks, and then next week we can work on replicating some of that stuff. And that's all. That's